Hello, my name is Cassidy Danyko. The paper that I reviewed is COX-2 gene dosage dependent defects in kidney development. This was done by Slattery P. et al. For the American Journal of Physiology regarding renal physiology. It was a study done recently on March 16, 2016. This paper investigates the cyclooxygenase or COX proteins. These promote inflammation by promoting prostaglandin and prostanoid synthesis. They are involved in converting arachidonic acid to prostaglandins. There are two COX isoforms expressed in the adult kidney. COX-1 is highly expressed in the collecting duct. It provides prostanoids involved in saltwater retention. Knockout of this gene does not lead to impaired kidney function, so this paper investigates COX-2 isoform. COX-2 is expressed in renal medullary interstitial cells, the ascending limb, and cells associated with the macula densa, which is involved in salt balance. Some of its particular functions found previously are that it promotes this prostanoid prostaglandin synthesis that's important for renal development. Additionally, they found it's important for regulating renin secretion, sodium excretion, and regulating the blood flow. They found that increased COX-2 expression led to high renin levels that led to vasoconstriction that can help return the GFR to normal, which is important for the kidney. Other studies have found that COX-2 plays a critical role 4 to 8 days postnatally. During this time, they found increased expression of COX-2, and then they found that inhibition impaired nephrogenesis. Additionally, inhibition during gestation did not affect kidney development. Researchers have performed studies on mice in which they block the expression of the COX-2 gene, called a knockout. In these studies, COX-2 knockouts lead to impaired kidney development, and this would include hypoplastic or underdeveloped glomeruli, which is not a good thing because this is the site of blood filtration with the Bowman's capsule. Additionally, they've found cyst formation in nephrons, cortical thinning, and poor tubule differentiation. Also, they found that the knockout mice have kidney insufficiency, and that's the function of the kidney. They find that there are high creatinine levels and urea levels in the blood serum, and additionally have found impaired filtration through the kidney. Other researchers have used gene dosing to better understand COX-2. In this, they can partially inhibit the expression of COX-2 by using an inhibitor. Under 90% suppression of COX-2 gene expression, similar kidney impairment was found to the knockout mice, but whereas in 70 to 90% suppression, there were minimal signs of renal dysfunction. In utero, COX inhibition had led to many kidney defects, including the incomplete. So the questions in this experiment are, what are the effects of complete COX-2 knockout and partial COX-2 knockout on kidney development? And this, they're wondering, are two functional COX-2 alleles required for proper kidney development, or can one functional allele in like a heterozygote compensate for the deficiency? So the number of functional alleles will affect the degree of renal impairment. And they think that there will be impairment in those completely knocked out more so than those partially. The other question they have are, what are the effects of pharmacological COX-2 gene dosage by inhibition? They figured that low doses of the inhibitors will show slight renal impairment, whereas high doses will have even more renal impairment. The researchers examined kidney development in knockout mice with COX-2 positive, meaning two functional alleles, positive negative with only one functional allele, and complete knockout. To ensure that the partial and complete knockouts were successful, they performed semi-quantitative PCR and found the partial knockout mRNA and protein levels were accurately 50% those of the wild type. Additionally, no signal was seen for the complete knockout. Then, for the second part of the question, examine kidney development in mice treated postnatally with SC236, which is a COX-2 inhibitor. They had a low. When the researchers examined the kidney morphology of P21, that's 21 days postnatally, COX-2 knockout mice, these knockout mice, the complete knockouts, worse than the partial knockouts, had reduced glomerular size, as in the diameter, and that's the where the little circle is pointing to. And they also saw some cortical thinning, which you can see with the bigger arrow. By staining the kidney samples with Sirius red, they also examined fibrosis and sclerosis. They found that the complete knockout mice exhibited more staining and therefore had more fibrosis. The partial knockout also had more fibrosis, as seen as the partial more redding. On P21, kidneys were removed from COX-2 knockout mice for histomorphometric analysis. Now, they found that the diameter of glomeruli and cortical thickness was smaller in the knockout mice compared to the control. As you can see in graph A, the wild type has a larger diameter than the COX-2 partial, which has a greater diameter than the COX-2 complete knockout mice. The same trend is seen with a corresponding decrease in cortical thickness. They used the distance of superficial glomeruli to the cortical edge to estimate the subcapsular cortical growth. The null mice had the most cortical. Additionally, the complete knockouts showed more superficial glomeruli growth. This indicates that many of the newly formed nephrons were in maturational arrest. Through further investigation, the newly formed juxtamedullary glomeruli also showed reduced diameter in both the partial and complete knockouts compared to wild type. They wanted to further understand what else is going on during the knockout. They added 2 controls to better understand the consequences of COX-2 knockout. They examined the expression levels of MPGES1 and PGIS, which are involved in prostaglandin synthesis. Interestingly, they found higher levels of expression in the complete knockout mice, and MPGES1 was also higher than wild type in the partial knockout. The role of these is still elusive. They also measured the prostanoid synthetic capacity of the kidneys. They added additional arachidonic acid to the kidneys and measured the amount of prostanoids produced. They found prostanoid synthesis capacity was reduced due to COX-2 knockout. You can see that there at the in graph E that reduced PGE2 and PGI2 synthetic capacity in both the knockouts compared to wild type. The renal defects of the knockout mice were attempted to be mimicked through the high and low dosing with SC236 COX-2 inhibitor. They examined urinary excretion rates and kidney development. Those treated with the low dose of inhibitor exhibited reduced PGE2 excretion rate, which indicated less prostanoid synthesis. They also had reduced glomerular diameter and cortical thinning, similar to those knockouts. The high dose ones had a greater degree of these renal. In order to determine the long-term effects of the knockouts, they examined kidney function of P90 mice by analyzing blood samples for urea and creatinine concentrations. This is a measure of the kidney's efficiency at filtration. Serum creatinine levels were higher in both the partial and complete knockout mice, whereas serum urea levels were only high in the complete knockout mice. Therefore, kidney function is impaired. Additionally, they examined the long-term effect of the gene dosing. Gene dosing led to a rise in serum urea levels, but did not affect serum creatinine levels, although there was a greater range in this data than the control, with a few points that are rather high. Overall, the researchers found that COX-2 gene requires both alleles to be functional. Therefore, a loss of function defect in one allele is detrimental to renal development. Additionally, the renal defects of partial and complete knockout mice were successfully mimicked through high and low dosing with SC236 COX-2 inhibitor. Therefore, renal development can be inhibited pharmacologically. An intact COX-2 prostaglandin system is required for renal development. So they found that COX-2 is important in nephrogenesis, the maturation of nephrons, post-renal development, and kidney efficiency in the function of the. This research clarifies the consequences of non-functional alleles regarding COX-2, so we can better understand human COX-2 polymorphisms if we can catch them. Additionally, we can better understand how to avoid nephrotoxicity when it comes to COX-2 inhibitors. Some limitations of this study were during the gene dosing, the SC236 inhibitor was actually not that selective for COX-2, so it's hard to separate the COX-2 and COX-1 function. Additionally, why did the knockout mice have higher PGIS and MPGES1 levels? So they're a little elusive about this feedback mechanism about what makes these levels higher in the knockout mice and how this is related to the defects. Additionally, they had inconsistent blood pressure results, so they can investigate other roles of COX. My first question is, what could be the danger of NSAIDs, non-steroidal anti-inflammatory devices or drugs? What if they are non-selective, and what if they affect COX-2 in the kidney? My second question is kidney impairment. I kept talking about this. What could be the consequences of reduced glomerular size and cortical thinning?